Thank you all for joining today's JETZON Open Call. As ever, we have two excellent speakers. And without using up further time, I'd like to go to our first speaker, Manu Prakash, who's going to talk to us intriguingly about the hidden comet tails of marine snow. So, Manu, over to you. Thank you.
All right, I'm assuming you can all hear me. I'm going to share my deck. And I'm assuming you can see the slides at this point. So thank you for joining in. I am in India right now, so it's pretty late down here. So I'll try staying for the whole call, but sets of questions folks have, please feel free to reach out. My name is Manu. I'm a faculty at Stanford. And for the last, I would say, 6 or 7 years, we have been thinking quite a lot about how to directly make measurements, especially of the microhydrodynamics of marine snow, and overall, more broadly, how to think about new class of instrumentation that's associated with carbon-related measurements in the ocean. I'll tell you about one story that we just recently published. This is work with my postdoc in my lab, Rahul Chajwa. And one of the exciting parts is Rahul is also in the job market. So if any of you are interested in these sets of threads. I think the first thing I was going to mention is a lot of the work that I'll mention and just the history of the instruments that we've been building is actually an effort from a larger group of students and people and postdocs in the past. So I'm not going to get a chance to acknowledge many people, but I'll mention a few sets of folks that are involved in the specific work. But for the larger context of instruments that we build, you can look at some of the work that I'll describe in the very end. There's also a fun perspective that Kale and Leon wrote on our work. So if you're interested in kind of getting a gist of the summary, you can also read that. It covers a little bit of the broader perspective and puts some of the work that I'm going to talk about in a broader perspective as well. Okay, so I think kind of with that as a background, let's just start with something that we all love. One of the things that I often, when I go to sea, is I realize you know, of course, when we think about the biological context of what happens in the ocean, is these are very large systems, and without the set of measurements, the complex details of what is at play is harder to anticipate. The story that I'm about to tell you today is something that without observational power, we would have never predicted. I mean, of course, ocean plays an important role in carbon sequestration, and especially we will focus on primarily the biological pump. And the thing that we had been thinking for a while is really this idea that, you know, we have some amount of pieces of the puzzle in how biological and especially more of a sedimentation-based pump actually works. But when we started thinking about this problem 5 or 6 years ago, we realized that direct measurement of sedimentation dynamics of marine snow were actually either very sparse or almost missing. And we realized that we have to make these sets of measurements at sea. Although there are sets of model systems people have been thinking about, much of the complexity of these particles as they form can only be recapitulated out at sea. So that was the framing and the perspective of how we got started. I think we come from a very microhydrodynamics and physics and biological background, and we started realizing that direct measurements would actually be a valuable framework to bring to the table, especially if we care about building broader sets of laws of sedimentation in this space. Many of you have heard about the Martin's curve in general and kind of thinking a little bit about how these sets of parameters are at play in how flux changes as a function of depth. But I think one thing that we found missing in starting to think about these sets of threads, and which has been highlighted in much of the literature, has been how does microbiology and the biological context of this living ecosystem that sinks to the bottom of the ocean, how does that play out in the sedimentation dynamics directly? And that's what we are going to try to address. You know, as a phenomena, an incredible number of things are happening. So if you kind of look at this picture that I've drawn here, all the way from what's happening to bacteria being able to find these sets of objects, the kind of organisms and cells that are composing and making marine snow, the length scale, which is in this low Reynolds number regime. But these sets of particles are actually sinking all the way to the bottom of the ocean, which is this astronomical set of length scales that are involved in this problem. And that requires new measurement techniques to be able to capture some of these length scales, which is this multi-scale measurements are always difficult and hard. Simultaneously, one thing that I just want to mention on the side is, of course, I will mostly talk about marine snow, but there is an incredible dynamics of the living ecosystem at play itself as well. And the behavioral context of them also matters significantly in terms of how carbon flux passes through many of these sets of layers, because many organisms, we realize, choose to be at a specific depth of the ocean. And I want to just give you one example to drive this point to home, which is, let's look at what we observe and think about as non-motile organisms. So you're looking at a few diatoms and coccolithophore. You can measure density of these sets of things, and you could think about, oh, wait a second, I could, at least in this case, estimate what the sedimentation dynamics of it would be. And you would be fair to assume that, but it actually turns out even that is wrong. What we assume these sets of organisms and how they would sink to the bottom of the ocean is actually dramatically controlled by these organisms themselves. And I just want to give you one example of this. So first of all, when you think about sedimentation, there is a classical law that you can apply, just the Stokes law, in terms of the density difference of an organism. So for cytoplasm, it ends up being 5 to 10% greater than seawater, and then a square to the size of the organism. This is what we have often thought about as sedimentation. But what I'm here to tell you today is that living organisms literally have a mind of their own, and they can do sets of things that are quite surprising. So first of all, everybody knows kind of the context of the 1D stratification in the ocean. We will talk about that stratification quite often. We got into this problem because we were very interested in starting to observe single cell and these sets of particles sinking over long distances. So the only way to do that is if you have an object that's 100 or say a millimeter, 100 microns or millimeter in size, sinking meters or tens of meters or hundreds of meters, is to build a very tall microscope. And we actually did build very tall microscopes. You're looking at an instrument that we built some time ago for tracking parasites traveling a couple of meters. And then one day when we were packing for this trip to Madagascar, we had an aha moment in the lab where I realized the fact that if you take a very long tube, and if you connect the two ends of that tube, you have built an infinite column of water. And that idea led to an instrument that we now call gravity machine, or essentially more technically a scale-free vertical tracking microscopy, that has a capacity to observe a single object either sedimenting or a single cell traveling across hundreds of meters or kilometers for that matter. And the idea is very simple. This is the first paper we published with Deepak and Hong Chuan, two of the graduate students in my lab. The idea is very simple. Could you build, using feedback control, a kind of a hamster on a wheel? Except the hamster in this case is either a marine snow particle or essentially a single cell. What you're looking at on the right is one of the instruments. It's the size of a dinner plate. It's made out of glass. And then there are 2 points at 3 o'clock or 9 o'clock in which we can cause, as the organism or the particle is sinking down, we can spin the wheel up. If the particle is going up, we can spin the wheel down. And with feedback control and machine learning, we can essentially get real-time control that you can observe an organism or a particle sedimenting at subcellular resolution, traveling kilometers. The way that's possible, so that's really kind of one of the framework. There's lots of papers that we have broadly published on this method, but I'm just going to show you kind of the intuition for this. So what you're watching here is a coral larva traveling hundreds of meters in this wheel, And as you see that this coral larva is climbing, the wheel is turning down. And this is all done hands-free using machine learning. So we are detecting the organism, and in a 10 millisecond loop, we are essentially responding to make sure that the object remains in the field of view. Now, there are a lot of tricks to the trade. First of all, temperature control. We do light control in these sets of systems. Now we do pressure control. There are new cues we can introduce. I'm not going to talk too much about that, but one of the frameworks is that these machines keep getting more and more complicated over time. And for the folks who think about the hydrodynamics of these systems, you can think of it in the context of as the walls travel up and down, they essentially have a momentum that they transfer. And that momentum is what leads to be able to keep an organism in your frame of reference. Now, we have done sets of scaling laws on this to make sure that we are not inducing any velocity to the object, and calibrated these sets of systems to make sure that what we observe is exactly what the sedimentation dynamics of an organism should look like. We've actually also done this in the context of shear and torque, which is important for living organisms. Much of this is published, I think, and it's just valuable to understand that we are still in a low Reynolds number regime. This would not operate and work if the system has inertia. And there is this bounding box for velocity that we can track, that velocity doesn't introduce any inertia in this wheel, which is why we can be faithful for these sets of observations. So let's just watch some of these things. What does it look like if you can sync effectively in the frame of reference an object that's falling? So I have a few sets of candy here that's falling, completely inanimate object. And very soon I'm going to turn the machine on, and you start seeing that suddenly now the candy that was falling is now fixed into the frame of reference because I've locked in on one of these particles. And although this is floating in your frame of reference, you're actually technically sinking at the equal speed. And from these sets of things, I can figure out the density of every one of these particles, look at interaction of these particles, look at dissolution of a given particle, and really watch the biology, which is really the most interesting part of these sets of organisms and objects over long periods of time. So just to give you a context of what a dissolution would look like, to the right is a piece of sugar that's sinking. So if any of you are having coffee right now, as I am, I'll play that one more time. The moment you drop a piece of coffee, you can see that these plumes of sugar molecules are essentially dissociating, and then this sugar cube is dissolving. But for the first time, you can be in the frame of reference of sinking, and you can observe a particle for long periods of time, literally for the first time. So what happens when we start putting living organisms? So this is an example of a larva that we study quite a lot. This is Pteria miniata. Again, the same framework. In your frame of reference, it looks like as if this object is literally floating, but this movie is a climb of this 500 micron object over roughly half a meter. And so what's valuable is that you're getting cellular and subcellular resolution dynamics of objects that are traveling over long distances. Now, of course, objects don't just do very simple trajectories. Here is a much more complex behavior of a sea urchin larva, and you can start seeing very complex trajectories that are played. One of the big surprise that we found is as we map behavior out in the ocean, we ended up discovering for roughly a data set of roughly around 600 species that we have studied so far in this framework, almost all of them have a capacity to detect gravity. And then they are positioning themselves actively in the z-axis. So the object that you're looking at the right is the track of it is unbounded in the xy plane, but it chooses to preferentially travel only along the z-axis. There are lots of reasons for it that we can talk about, but behavior, and especially gravitaxis as a behavior, is universal in the tree of life, at least in the planktonic world. Now, I'll give you an example of something that is very counterintuitive, and then I promise I will talk about marine snow for the rest of the time. This is unpublished work, but it's important to highlight that objects that we have thought that are non-motile actually have motility. And what do I mean by that? This is a cosmodiscus, it's a diatom. And you can see I'm letting it sink in the machine. And within 100 milliseconds, it changes its density and goes from a perfectly sinking particle to perfectly stationary. There are no walls here. The object, the cell itself, is choosing its density and modifying its density in real time. And then 100 seconds later, it say, no, I don't like it here. And it starts moving again. And it's just absolutely bizarre behavior. But literally all of the sets of cells that we look at as when they are living, they have the capacity to modulate their density in remarkably complex ways, which sets to question many of our passive models that do not account for the dynamics of these organisms. And again, what sets of sensory cues might be controlling these sets of phenomena? Similarly, we've been studying buoyancy and dynamics of cells as a context of cell cycle. I don't have these videos here, but for many dinoflagellates, we have shown that density regulation is a function of cell cycle. The sets of tools allow you to do PIV and sets of very close measurements that would not be possible otherwise. So let me now switch to marine snow. It's been a joy to take these sets of instruments out to sea. In the last 5 years, we've done around 17 expeditions. I'm going to focus on telling you data just from one expedition, but we are slowly starting to release all of the data sets so then people can actually play with it. Of course, much of this is possible because of phenomenal sets of collaborators that maybe I'll mention in the very end, but this is a very large network of collaborators that we work on. And anybody who's interested in kind of really being able to measure microscopic details of this, we're very excited about collaborating. But let me just show you what happens when we take these instruments to the ocean. All the data that I showed you was also from the ocean, but the first thing is doing microscopy at sea is hard because your platform is not stable. It's wobbling all the time, so we have to account for that. We do that by making these gimbals. These are gimbals that allow us to passively stabilize the instrument. So you can see it's canceling the X and the Y tilts passively. This is an older version of the instrument. Because of this, we can really do very long-term measurements. And again, now transitioning to marine snow, this was an object that we wanted to study for a long while. And it's a little bit like as if you are in a tiny little submarine and you could watch one particle travel over long periods of time. Most of you know the context of how marine snow is captured, but just for some of you who haven't thought about it, this work that we did, this is with a collaboration with Ben's lab at Hui. They've been using sediment traps. All the data that I'll show you is for 80-meter traps, but we do the same thing for 200-meter, 500-meter. We've been thinking quite a lot about 1,000-meter traps. And of course, this is done in a Lagrangian context off the Gulf of Maine. So let's just watch what marine snow looks like in this context. I've just chosen a random object here. These sets of particles were collected at a given depth, loaded in the wheel very carefully, and we've done lots of work in making sure that these sets of things represent the true activity. And you start watching, and you can then lock into one particle and observe it if you want for, say, 12 hours. There is a lot of phenomena that we have discovered. I will just focus on mucus tails, but we've been studying microbial interactions, feeding dynamics, fracture, many sets of phenomena that are at play. I'm only mentioning one particular phenomena that's very important. So the first thing we realized is that we should build a database, because all of you know these types of particles, and I'm only considering here the classical marine snow particles that form because of aggregation of sets of ecosystems and plankton. So this data set doesn't include fecal pellets, although we've done a lot of work on fecal pellets. So on the left is images in the right orientation of how those particles were sinking. And then I'm just going to show you one quick movie for what that looks like. The scale bar there is around 200 micron. We can do around 60x objective, so roughly a resolution of 700 to 300 nanometers as kind of a highest resolution we can do in a stable manner. Of course, all of that data is collected at sea. And I'll show you the data one at a time, but I just want to first have you stare at these two pictures. On the left is the images of marine snow particles, and on the right is the average sedimentation dynamics of, say, all the way at the bottom is 200 meters per day. And you start seeing that although I've organized on the left particles as a function of size, there are lots of very small particles that sink really fast. There are lots of very big particles that sink very slowly. So kind of our classical ways of thinking about sedimentation doesn't have to be true. Now, this is true sedimentation because in sedimentation, the object actually chooses its own orientation as it's sinking. So this is just the distribution of those sets of velocities. And you can see a classical Stokes law doesn't really explain the spread. And of course, lots of people have been thinking about it, but the big puzzle has been, where does that spread actually come from? So now what we are going to do is really focus, and I'm going to zoom in on one of these particles to show you that there is a lot of fluid structure interaction in these systems, and there is a hidden degree of freedom that was missed in the past sets of observation. And that hidden degree of freedom is actually mucus. So let's look at this one particle. And of course, I could spend an entire evening showing you many of these data sets, but this is just a representative data set. And we will just watch this carefully. So I don't know what resolution you can see this on Zoom, but notice the flow field. By the way, these little punctae or little stars I have seeded the field with tiny little particles that allow us to also visualize in 3D the flow field around the sinking particle. And since this is the most important movie, I'm going to play it again. And notice very carefully, right behind the particle is this kind of massive lump of a fluid that's traveling with the particle. That makes no sense if you were to think about Stokes' law, because usually this fluid should not be moving, and it actually turns out that is the signature of a mucus parachute, or what we have termed as comet tails of marine snow. Now, it's important to understand that these are not chemical trails. This is actually a physical mass of polysaccharides, and I'll show you in a little bit that this is what this is composed of, that's trailing behind, creating a new term for drag. And so when you look at this in PIV, you can see this massive zero flow zone right behind, which is acting like a parachute. It's a physical parachute behind this object that both changes its net density because mucus is lighter, and at that same time changes the physical drag. So let's just look at this data more broadly. And what you notice is that this is a signature that exists in almost all of the particles. It's not just one or two particles, but significant portion of particles have this. And when we correct for this, we actually find that the effective size of marine snow is not just what the visible part is, but this invisible part. And what this leads to, just as an overall framework, is reduction by a matter of half to the overall average residence time, for example, during sedimentation. Now we can stain these particles. We can show that most of this is really explained by TEP. And one of the things that I'm not going to have time to cover in this talk, because I'm going to run out of time, is we have built a new mathematical model that allows us to account for the sedimentation dynamics and sedimentation stress as a function of sinking, and be able to calculate variety of properties of this mucus directly from these sets of data sets. And now this allows us a modification of the Stokes law that then we can fit to demonstrate how sets of particles actually sink as a function of time. So one of the things, an important lesson to take away from here is the residence time of all of these sets of particles is doubled in this particular expedition. And this leads to an error in how we have been thinking about sedimentation. And we've been thinking about what that total error might be, but this doubling enables microbes to remineralize this mucus. And in some of the perspectives that Kale has written more recently with Lyon, that roughly accounts for around 100 gigaton of a discrepancy if you were to calculate these types of frameworks purely using a classical Stokes law. And now this is the modified version of the law. And we are starting to build this framework to fit this to larger sets of other data sets, and also expanding these data sets in different parts of the ocean, especially in different sets of plankton communities and parameters that lead to this production of this mucus. Because I think one thing that we don't understand as yet, although in all the 5 expeditions that we have done this, we see these comet tails as a key signature in almost all the data sets that we collect. We don't as yet know, are there other sets of parameters, for example, stress of these cells, temperature, pH, how these knobs, and especially because these are microbial mucus mats that are generated, how they change as a function of time. Now, I think the last thing I want to mention is we also do a lot of imaging, and especially volumetric imaging on these systems to be able to understand. And one thing that we have demonstrated in this, although for a long time people thought that marine snow was porous, we don't see any evidence of true porosity. It appears porous, but these pores are filled with mucus and it doesn't allow fluid flow, although it does allow for a lot of bacteria to take residence in there. And in fact, in some of the most recent expeditions, we are now able to track during sedimentation dynamics, individual bacterial arrival and leaving in the context of remineralization. So the movie that you're watching on the right, you're watching a single sedimenting particle, but in the flow field are individual bacteria that are sensing this sinking object and landing on it. And we care about this in the context of remineralization. I know I'm already out of time, so I'm just going to conclude that this has allowed us to think about a new way for sedimentation dynamics, which is a physics-based approach to understanding the biological pump. This is a fairly large discrepancy in how people have thought about this before, and we are now building larger scale models and trying to incorporate these in classical models, especially also making this data set available. And I'm going to end here by saying this has been a fantastic collaboration with Kay's lab, Ben's lab, and one of my graduate student, Ellie, and the postdoc that I already mentioned, Rahul, who's been one of the champions in this project. I'll end here and I'll leave one picture in the background for if some of you are interested in frugal oceanography. We've been building lots of tools, and one of the tools that we have released is called Planktoscope. This is for open source microscopy for plankton. And if you're interested in it, you can go to this website called planktoscope.org to learn much more about it. And I'm going to end here, and I still have a few minutes so we can take some questions if there are any.
Thank you. Some extraordinary stuff there, Manu. Absolutely extraordinary. Thank you. So I'm sure we must have lots of questions, but we're going to need to get 2 talks in. But maybe if we can have at least one quick question if somebody has one. I think may have seen one in the chat. Was that a question, George, or was that more of a comment? I mean, feel free to ask a question if you'd like.
More of a comment than a question. Just great work, Manu. Love seeing this following of small particles. We could follow large particles in the field, of course, but not these tiny ones that you're following.
Yes. Yeah, and I think kind of one comment that I would love to talk about is for people who have other in situ data sets, we are starting to corroborate these sets of, so I'm super excited about a new instrument that we are starting to build with some collaborators, including some people in this call right now, because one of the goals is to build continuous framework, and I've been very excited about what has been done in the past and making sure that we have a continuum of data sets, because frankly, we need these data sets to be able to build better and better models. Otherwise, sometimes we can be blinded, and especially when biological parameters are at play. So I am super excited about also incorporating some of these data sets with the in situ measurements that have been made.
Fantastic. Thank you. That's an excellent way to round off as well. So unfortunately, we have to move on to other things now, but thank you very much once again, Manu. If people have any questions, I'm sure he'd be delighted to hear from them. Either type them into the chat or feel free to email him, and we'll be putting the talk up with a link very soon, hopefully.